some network issue once again. I'll be joining in. Yeah. One second, I'll just it for 5 minutes in this particular slide, and then before going into this particular slide, and then we can start, right? So I'll just wait for 5 minutes for you all to have any particular questions, and then we will move on, okay, with the protein microarray. So in 5 minutes, you can ask any of the questions that you feel. Okay, please feel free to ask at the end in the quiz format, as I have already mentioned. We will be having a particular, what to say, a particular uh, question discussion for your uh, towards your solving of the assignment. Now you have a chance for 5 minutes. Please ask the question. Please feel free to ask any questions till now of the protein technology that we have talked about, and then we will shift our gears towards protein microarrays and their understanding. Right? So we will be resuming with 5 minutes. So when you are ready with your questions and we are able to discuss it, and lastly, we will be having like a quiz-like discussion of your questions towards solving your assignment. Okay. Yeah, I think all of you are getting ready with your questions, and we will discuss it before moving into the next section of protein microarrays and the interactomics. We have covered a lot in the proteomic technology, and we will be moving further ahead, right? Followed by a discussion on quiz to get an understanding that how we can take it forward, how we can, how it will be helpful for you on the questions in your assignment. Right? Yes, so please come up with a question. We have covered a lot, right, about your particular procurement technologies, and then we will be moving forward for the interaction-based technologies, right? And then we will be moving towards the microwireless and other interaction technologies, and then we will have quiz like discussion for solving your assignment. Any questions? We will wait for 5 more minutes. Then we will move our gear, shift our gear towards the microwireless and much more interactive network. Any more questions? Any question? We will shift our gear to protein microarrays and motor interaction tools. Please feel free to ask. We have covered a lot in different proteomics technologies. Please feel free to ask the question, right? We will be discussing some questions, right? Related to solving of your that will help you on solving of your assignments in a quiz kind of format. Please participate in that. So we will wait for 5 more minutes and then we will shift our gear. So final questions, any questions that you have? And finally, so we will be completing the next phase of microarrays and other interaction tools, and then we will quickly move through to the discussing some of the questions that will be helping you and discussing those to help you with your topic assignments. Okay, 5 minutes more to get the questions. Any doubts? Any question? 5 more minutes. Okay, so 3 more minutes and we will move on our gear towards the major findings, what the interactions or the protein microarrays, and we will go towards it, and then finally some question answers in this format to get help you in the solving of the assignment. Okay, I hope here. Any questions? We have covered a lot from the concept from the week one of the interactomics. Okay, one more minute and we will start, and we will see that how much we can, how fast we can cover and then discuss. I think all of you are waiting, right, for getting discussing the questions that will help you to solve your assignments. Same questions we have covered a lot from your first topics to try to find out the ways and manage it, right? Yeah. So let us move back. I think we should be moving forward. I think also to get idea that how this particular some of the questions to practice for you all to solve your assignments, right? Yeah. So protein microarrays are used for like studying your protein antibody, protein interaction, etc. So like immobilization of your particular capture agents, the sample incubation, then washing and detection. So here I would like to say that you have like covalent interactions, etc. etc. So if you are doing it by adsorb, so that is not a very good way. But the immobilization can be like through covalent bonds and other interactions. So that you have to keep in mind. So this is one of the papers that have talked about the like the autoantibody profiling of the plasma serum samples to identify the biomarker thinking. Uh, well, it was there, right? So because you can easily go and easily find out, apply this kind of techniques in early detection of cancers and other ways because it's very, very important. It's very, very uh, much required, right? So it is very much required to immobilize those agents, incubate those samples, and then detection. And using these simple technologies, you can screen a lot of proteins, across 20,000 proteins of present in your body. And through this, you can get the expression intensities and then you can identify those markers and you will be getting it. So this kind of profiling and this kind of technique, uh, so this can be taken forward. So this you can take it, what I can say, as a forward, right? So you can take these things as forward for a variety of applications like early diagnosis, as we had talked about, right? That how this kind of particular, what I can say is that this kind of particular strategies or the procurement technologies are can be used for diagnosis purposes or treatment response of the cases of this. This is early diagnosis, one of the major application can be the antibody profiling. Okay, so these are again level-based techniques because antibodies are used. Now here also in the protein-protein interaction, we have the level field technologies like surface plasma systems, where ligands are immobilized on the surface of a gold chip and analytes are flown over the ligand to characterize the index. If there's a interaction happens, so it will changes with changing the refractive index. An increase in mass associated with this event cause a proportional increase in the refractive index. So it will have association, equilibrium dissociation. So this is your level free technique. So again, the interactions can also be level based or level free. Like microarrays are your kind of level based techniques, but here it's a level free techniques. Now, if we do SPRMS, so this is a case study you can thought of like kinetic characterization and identification of the epitope binding by SPRMS. So what is that? So, chip, SPR chip, and then you are doing it so well suited for samples having your multiple components and allows to carry out affinity based isolation of the epitope, kinetic characterization and identify them, and you can have like off chip and on chip configuration. So now if we move on to interactomics, so this is what, so this is a protein and this protein will be interacting with this particular protein. So interprotein, uh, this thing does not only belongs to one particular thing, right? We can talk about like the protein. If we say like the protein functions in unison, it is not that so simple. Understanding protein function also plays role in the kind of environment it is, a kind of the interactions, the surrounding molecules. So everything is dependent on to understand the interaction and the physiological movement and everything. So proteins, what it happens is that it interacts with other proteins and other biomolecules for a variety of functions, including the protein transport and to determine the roles of these uncharacterized proteins and then regulation of the protein activity and to construct the network of protein interactions. So there are different methods of interactions like transient, permanent, weak, strong, molecular, heterolegular. So significance of PPIs that cellular processes and functions are regulated by this kind of PPIs. Signal transduction, cell growth and regulation, protein transport, biomolecular interactions, alteration of enzyme cascades, physiological processes, and it's also like you can talk about like governed by the electrostatic interactions, hydrophobic interactions, van der Waals interactions, and hydrogen bond. So this is pretty important. So this kind of protein-protein interactions are quite vital in nature because it's all about like the cellular processes and function, signal transduction, cell growth and regulation. Then you have like the protein transport, then biomolecular interactions, alteration of enzyme cascades, physiological processes. And so you can see like organ network, cellular network, molecular network, genetic network. So studying protein interactions, right? So that's what the course is all about, interactomics, right? So all the interactions, if we try to find it out, right? So if we try to find it out, all the interactions, then what we are going to do, right? So if we talk about the studying of the protein interactions, then one thing that we should know, or we should keep in mind, that there are two approaches. One is a conventional approaches, another is a high throughput approaches, right? So conventional approaches consist of is2 hybrids, affinity chromatography. So affinity means you know that on having one particular affinity, immunoprecipitation using antibodies and precipitation, and high throughput arrays like protein microarrays, which we have talked about, level based protein, level free. Okay, so I think all of you can take 2 minutes again to think else. I'll just be back within a minute. Okay. Yeah, sorry. So we have done that. So I think all of you have realized some questions, right? So this is the conventional approaches that we take, right, for our going through the technologies like is2 hybrid, affinity chromatography, immunoprecipitation, but whereas high throughput approaches tend to take like protein microarrays and the level free technologies. So what is immunoprecipitation or co-immunoprecipitation? So this is a very traditional way of understanding that how you can identify. So for example, if you are willing, so you can take the cell lysates, right? Like the total proteins that are mixed, add antibodies, right? And incubate. So this antigen antibody complex, so for example, add protein beads and incubate it. Then this beads complexes with other proteins like which are all co immune precipitate is there. It will be washed and eluted. So this antigen antibody beads, which no other proteins will can be used for like Western blot staining technique, mass spectrometry, right? So this comprises of like a selection of a suitable antibody, binding of antibody to protein, elution of the antibody and protein complexes, followed by purification and downstream analysis, right? So this is how you can get it. So non-denaturing conditions and denaturing conditions. So this will be the non-denaturing, then we will get the denaturing and try to understand the protein-protein interactions using antibodies or immunoprecipitation. So what are the merits and demerits? So these merits include like the natural interaction. So this can help us to get the actual interactions. Then large complexes can be purified. Proteins in native state interactions possible. Deep edits are like non-specific interaction, direct indirect interaction or indirect interaction that we don't know, and expensive. So why this demerits we are talking about, like direct interaction, because the protein is there will be many proteins coming up with one antibody. If we pull out one protein, now that protein is mapped with the second protein is directed with the third protein, fourth protein is in fifth protein, and so on. But now this first protein and fifth protein is directly interacting or the because you can understand this is a chain, right? So if you have for instance, if you talk about like this is a chain of proteins, then what this particular chains sorry. So if I just try to, so this is for example one antibody, right? It is pulling this particular protein. Now this right protein, this protein, this protein, this protein. Right. So now this protein and this protein is directly interacted, but this and this is not. So this is not at all possible. So that's what we talk. Then non-specific interactions can be possible and expensive. So if the proteins in native state, interaction possible, the large places can be purified. Okay. And then actual interactions can be identified. So this is merits and the demerits. Okay. So this is what we can get. Okay, so these two hybrid systems if we talk about, which is a conventional method. Also, immunoprecipitation was the one. So this relies on the expression. So what happens that if we just change it to the letter for so it relies on the expression if a reporter gene activated by the binding of a transcription factor. So transcription factor comprised of DNA binding domain and our activation domain. So this is known as our bait and prey system. So query protein of interest. Library. So you have protein and there's a expression of protein libraries with our activation domain where your binding domain will be interacting with the protein library. So then it will be activated and it will try to generate, right? So it will try to express this particular protein and you to know that this proteins and this particular protein interact. So thus hybrid proteins are jointly expressed in this nucleus and the expression of the gene only possible if the bait and the prey interact. So right. So if the baits and the prey interact, so then only expression of this particular gene is possible. So this is the, so example, if you have the reporter gene with the activating domain, and so if this is the kind of libraries, protein libraries, if we adding it, so if this domain and this protein interact, so wait and pray, so it will finally transcription. So this is not interacting, and this is not the transcription, but this is interacting and getting the transcription. So this is how the yeast-2 hybrid systems work. So used for protein RNA, protein DNA, and protein-protein interactions, and it can be performed in a quite cost-effective manner. Not so difficult protocol can be used to screen large libraries. High false positives or negatives, and localization of proteins in nucleus is a must. Non-yeast environment, question mark, what would be happening? Pairwise interactions or multi-interactions. So then we have like protein microarrays, which comprises of like antibody arrays and protein microarrays. Major challenges are spotting the purified protein. So this is a bigger challenge. So that's why you need a robotic uh, arm and help, so that all the proteins of equal concentrations are uh, printed on each spot in a very good manner, right? So this is the very good science paper which shows that global analysis of protein activities using proteome chips, right? If we talk about types of protein microarrays, then we have like one, some direct labeling. So antibodies are printed on the glass slide, and proteins are directly labeled with dyes. So antibodies are printed on the glass slide. So just try to understand the terminologies when we're talking about direct labeling. So antibodies are printed on the glass slide, and proteins are directly labeled with dyes measuring fluorescence intensity. In sandwich immunoassay, intensity measured in secondary antibodies. So this is antibody, so primary, the secondary body. And the reverse phase protein arrays is that when we talk about reverse phase, that the cell lysate is printed on the glass slide, and so cell lysates are gets printed on your glass slide, and the fluorescence is captured through primary and the secondary antibody. So if we talk about some things, one is like the peptide arrays, then we have the NAPPA, and we have like multiple spotting techniques. So this one we can talk about abundance based array or the function based array. So the protein microarrays can be again divided into two parts. So protein expression and all what we are talking about. So this is belonging to the abundance based arrays. Whereas if we talk about the function based arrays are also possible. So high throughput with small amount of analyte, but protein purification remains major challenge. Now, these all are of the cell-based expression systems and all which we are talking about. Now, if we talk about like the cell-free expression system, right? Protein microarrays. So this produces structurally and functionally correct protein molecules. So it accomplishes protein synthesis by means of cell extracts containing all elements of transcription and translation, and some energy sources or modifications required for the expression of the systems. So now if we talk about cell-free expression systems, then again there are three types. Protein in situ array. So this is like PCR, DNA, and cell-free lysates, coupled transcription and translation, wash and pizza arrays here, NAPPA. Plasmid and cover entire array with the cell-free lysates, coupled transcription and translation, and in situ from mRNA array. So these are again three types when we talk about the cell expression. With NAPPA one is most popular and useful. Okay, so now if we summarize, trying to summarize, then what we can say is like proteomics, like omics-based technology, complementing genomics technologies, and helps in further assessment of the systems level approaches. So like different approaches are there. So antibody and MS-based methods and high throughput techniques, which we can talk about like the microarray based approaches. Okay. So again, 3 minutes we will discuss question. In the 3 minutes, you can ask if you have any query or any questions, and then we would be going to uh, discussion of the questions where we will be solving. So what I can say is that here we will be trying to solve. So I had, as I mentioned, that I will quickly run through the concepts because all the concepts you have already gone through the lectures in the week one. But I quickly glance through so that it's a complete revision you get. And now we will be discussing some questions in the quiz format, which will again help you, right, to solving your assignment in a quiz format. Okay, so again 2-3 minutes can take to frame any questions, and then we will move towards solving questions in a quiz format or discussion of those questions in the quiz format for helping you that will be taking forward towards solving your assignment questions. Okay, so 2 minutes to 3 minutes, and we will start again with the question. Okay. Yeah, so 1 more minute you can take, and then we will move towards the question that will help you towards the solving of the assignment. Let's see. Yeah, so this is where we will now look into the questions, right? So that will help you towards the solving of the assignment. I think all of you will be really happy that this image gives that you can solve all the assignments quickly. So you all can just scan this QR code, right, with your real name, and you can just take this quiz type, so it will be helping you to discuss, to revise the concepts, right, and then to solve the assignment. Okay, so you press enter. So the more the faster you place, you will get answer. So the question is that in the immunoprecipitation technique, which we had discussed, an antibody specific to the plate is attached with the English protein in the cell to form the complex immunogenic. True or false? That's a simple question that you need to answer. The answer is false. So antibody specific to the plate is not attached to the enriched protein in the cell to form the complex immunogenic condition. Right? Okay. Now question two. So there are 12 questions. Mahesh wants to identify protein coding genes in this experiment. Which of the following techniques can be used to identify the protein coding genes? Is it nucleus S1 mapping, full blotting, ORF scanning, or transposon hacking? So hopefully all of you know it is ORF scanning. In this way, we are now discussing the concepts, and that will also help you get your particular idea about the solving towards your assignment, right? Now we will move on. So question 3 of 12. Which of the following statement holds true for the GAL4 reporter gene is two hybrid system? GAL4 can be fused to the activation domain of transcription factor, can be expressed only if the tested protein interaction occurs. Fused with domain of a transcription factor requires the presence of histidine in the growth medium. What is the correct answer? So it can be expressed only if the tested protein interaction occurs. Good. Okay, so now we will be moving towards our next question. Our next question is, the protein microarrays are composed of thousands of discrete proteins and used for various applications, except for which of the following: biomarker discovery, protein body interaction, functional characterization, structural characterization. Which of the following protein microarrays cannot be used, right? So it is structural characterization of different biomolecules. Now, question 5 of 12. So the fifth question, the protein microarrays are classified into two broad categories: abundance based and function based, which we just now discussed, right? Is it true or false? So I think we had just discussed the concept, and also it has been quite explicitly discussed in our big one. So it is true, right? Abundance based and function based arrays. So question 6. Which of the following does not define the concept of personalized medicine, which we have discussed today itself in the beginning, using an individual patient's phenotypic information for these or are clinical markers, medical plan personalized treatment can be done in both prospective and retrospective way. In, so which ones? Using an individual's phenotypic information of his or marker, right? So next. Which of the following is not valid for the field of interactomics? It aims to identify the functional characterization, does not lead the characterization of new proteins, to understand the mechanism of regulate protein activity, develops networks of protein interactions, so does not lead the characterization of new proteins. So that's the right answer. Which of the following tends to form the strongest bonds in proteins? So is it electrostatic interaction, hydrophobic interaction, hydrogen bonding, or covalent bonds? Its covalent bonds contributing to protein structure. Which of the following is a high throughput technique to study protein interaction? So, which we already discussed: affinity chromatography. Is it protein microarray? Is it immunoprecipitation? Is it surface plasmon resonance? So, which of the following is a high throughput technique? It's protein microarray. Okay, so in cell-based versus cell-free arrays, which of the following is not a limitation of cell-based arrays as compared to the cell-free arrays? Protein stability, protein expression and purification, no technique like PCR or protein production, only pure protein is printed on the array. Only pure protein is printed on the array, right? Yeah. So we'll go to the next question. So in Napa protein array, how one can determine that protein produced from IBDT is producing protein which is functional? Test using anti-GST, test using IgG, test using blank spots, test using antibody kinase, right? The test using antibody for kinase. So well done to the winner. And now, obviously, I think all of you would also be able to solve your assignments without any doubt. For 2 more minutes, and then we can come to conclusion for this session and end this session. So, if you have any questions, right? If you have any further questions, and then we can end this particular session. And hopefully, you would be able to perform your well in your assignments for this week one. All the best for your assignments, and also you would be doing well towards your final exams as well, right? Any questions? 2 more minutes, and we will wait to take any further questions, and then we will stop the session for this week one, and we will meet again and in the same time. Okay. Okay. So I think then we can end today's session. So thank you all for joining, and hopefully you would do very well in your assignments and your final exams. So we'll meet next week again. Thank you. Thank you all.